AICT. An institute offers B Pharmacy, M Pharmacy in 5 specializations: pharmaceutical chemistry, pharmacology, pharmaceutics, pharmaceutical analysis, and pharmacognosy. The institute is also offering the PhD program, under which presently 85 members are on roll and total 56 members are awarded PhD degree till date. And IPT has a team of 15 dedicated, well-trained faculty who are ample national and international exposure. Institute has received several research grants such as UGC SAP, DST, FIST, and AICT MADROB. And our research activities are funded by several governmental agencies such as UGC, DST, DBT, and AICTE. And the department has several collaborations with national and international institutes of repute. In the last 25 years, more than 600 research papers have been published with a citation index of 31 to 322 and SAR is around 1 to 20. It demonstrates our scholarly influence. Till date, 5 faculty members are received the State Best Teacher Award and 3 faculty members are received the State Best NSS Program Officer Award. And during the 2019, our institute has bagged 42nd rank, NIRF rank among the pharmacy institutions. And the IPT is committed to cater the needs of the pharmaceutical academia and the industry. And hence, the present FDP was proposed in the area of artificial intelligence, which has an ample opportunities for the faculty and the students for gaining of the foothold in the rapidly changing scenario of drug discovery and development. So this is regarding our institute. And regarding the FDP, so artificial intelligence is a current buzzword and it is gaining the attention in all fields of the research, and there is no exception for the medical field. And artificial intelligence knowledge is becoming more essential when pharma industry is moving ahead towards the industry 4.0 concept. Moreover, artificial intelligence can upgrade and expedite the research and development efforts, and it diminishes the time and expense of the early drug discovery, making the process more beneficial. And to keep with the changing scenario, the objective of the present faculty development program is impart the current aspects of the applications of artificial intelligence in the field of drug discovery and development. In the present FDP, a total of 13 sessions were planned. Out of them, 12 are technical sessions and 1 is based on the stress management. And with the 12 well-experienced resource persons and covers the topics from theoretical aspects of artificial intelligence in drug discovery and development. And in these softwares, algorithms utilized to practical sessions of the handling those tools. This will boost the confidence of the participants to fuse the learning into their research areas. The research persons for this FDP are having rich experience in their area of research and have real-time experience of implementing the artificial intelligence in for drug discovery and development. Hence, exposing the faculty to with an interdisciplinary background and possessing the excellent research skills will instill the confidence in the faculty to venture into this area of research. So This is regarding the significance of our artificial intelligence. And all the best for all the participants. Make use of this FDP, and I hope that you may go with handful of artificial knowledge when this FDP is complete. Okay, thank you. Now I will hand over the session to Professor Shailaja.
Thank you, Sridevi. Good morning, dear participants and Professor Purachikodi, Madam, today's resource person of the present session. Madam, I am pleased to invite you to the AICT-sponsored online Atal FDP program. Dear participants, I am delighted to introduce you, Professor Purachikodi, Madam. Madam is currently Professor and Head of the Department of Pharmaceutical Technology, Anna University, Trichy. Madam has accomplished her bachelor's, master's, and doctoral degrees in pharmacy discipline. She has more than 25 years of research experience and teaching experience. Focused her research on computer-aided drug design and has developed a good number of novel heterocyclics such as indoles, thiazoles, oxazoles, imidazoles, and benzimidazoles. She also worked on 14 medicinal plants and has expertise in screening of bioactive molecules. So far, she has successfully guided 11 PhD scholars and some more are on roll. She has published 5 book chapters and 3 books. Her I10 is 31 Google Scholar from Google Scholar. Her H index is 16 and her Scopus index is 56. Total citations are 968 for Madam. She has a patent for her credit on isolation process. It's a process patent. Isolation process of a natural compound with anxiolytic and anticonvulsant properties. Madam has also isolated and retrosynthesized a natural compound from marine source. She has received project grants from DST, UGC, and some more from Tamil Nadu state government to do her research. She also received travel grant for visiting UK, and she has recently received INSA Visiting Fellowship, Jorhat, Assam. Madam also received distinguished awards and honors in recognition to her contribution in research and academics. A few of them are Fast Track Young Scientist Research Award she received in 2006, Young Women Scientist Award in 2008, Professor S.P. Tyagarajan and Professor S. Rajurajan Endowment Award for filing the patent, and she has, in 2011, she has delivered more than 40 invited lectures at international and national levels. She also received grants for organizing 6 seminars. Madam has organized and participated in several conferences, seminars, workshops, and faculty development programs. She played a vital role in publishing package of practices for 24 Android apps in Google Play Store related to safeguarding top 10 endangered plants identified by Government of India in southern India. She acted as a coordinator for COVID-19 National Bioinformatics Hackathon this year. She is a member in different professional societies such as IPA, Indian Science Congress, etc. With this brief introduction, Madam, I will once again welcome you to today's online Atal FDP program. Before Madam starts her presentation, I request the participants to type your questions related to the presentation in the chat box so that Madam will answer them at the end of the session. Now I request Madam to deliver her presentation on artificial intelligence for drug target identification and validation.
Can I start now?
Yes, madam.
Yeah. And thank you, madam. Thank you for your nice introduction. It is my great pleasure to get a great opportunity to speak among the professors and various experts of this Atal program. And I am Dr. Purachikodi, heading the Department of Pharmaceutical Technology in Anna University BIT campus, Tiruchirappalli. So my expertise, Dr. Shailaja has given all the details. So with this introduction, I would like to start my lecture. As he said, the title of the program, that is target identification and validation using the artificial intelligence. 1 minute. Okay. So this is a wide topic. That's why I have chosen a focused topic to explain you in detail. This is my research topic. I have completed this research at NIST, Jorhat in Assam in the last year during the INSA visiting fellow using the artificial intelligence technology. The title of the research is Identification of Hub Genes and Key Pathways Associated with Co-Infection of Urinary Tract Infection and Diabetes via Integrated Bioinformatic Approaches. Already, Professor Sridevi explained the need of artificial intelligence, how it is utilized in all the fields, and what is the impact of the artificial intelligence in the drug development field. So AI is a technology. It's a technology. We are trying to utilize the technology to develop the drug molecules and target molecules. AI technology is used to forecast the disease spread and to assess the corresponding disease. Used as a prognosis, not diagnosis, is a prognosis device. Also, AI-based data analytics and forecasting, which enables the medical professionals to understand the cellular, molecular functions, how the disease is processed, and what are the pathways are involved. So many information is concerned with we are generally telling us here, gene ontology. It's generally called as gene ontology. So where is the location of the exact location of the gene that is responsible for the particular disease, origin of the disease. That is a cellular and molecular, how it works and how it develops, how the disease is developed, and what are the mechanisms involved. So everything can be identified using the artificial intelligence. With the use of AI, more accurate and forecasting disease spread. We can, where it is exactly. So we can locate the disease spread and which appropriate medication for the disease spread and what type of treatment we can give. So everything can be analyzed and get the information from the AI. And very important thing for all the researchers, it scans the vast data of the medical literature related to genetics, proteomics, genomics, and to identify the new targets. Is a very interesting thing and very surprising also, because the early period, we are searching the information and literature in the literature, that is in the hard copies, in articles and in the library and visiting the library. And so many methods we have used. Now, everything is available in the online because of the advent of this artificial intelligence. It has the ability to find the hidden documents. We cannot, because lakhs and lakhs of informations are available. Every person cannot search manually all the informations. So the hidden documents and hidden information and unintuitive patterns in vast amount of the data in ways that no human can do, but this technology can do. AI represents a considerable promise. It has the potential to transform many industries, including pharma and biotech, particularly during the COVID period. IoT technologies are very useful for the development of the drug development process. And this technology has the ability to learn itself, to reason itself, and make correction itself. Is behaving like a human. And it has the ability to execute the task naturally associated with the human intelligence. That's why it is called as artificial intelligence. Its knowledge is almost similar to that of the human intelligence, like speech recognition, decision making, visual perception, and translating the languages. So that's why AI technology is very, very essential to in the field of pharmaceuticals, particularly to identify the hit molecules, lead molecules, final drug molecules. Similarly, on par with that, identification of the biomarkers and the development of the proteins. So the AI technology is very, very important and is a mandatory one for the development of the pharmaceutical technology field. So in the particularly in the field of pharmacy, not the pharmacy, in the R&D area of the drug development, what are the AI driven solutions? AI, artificial intelligence for target based and phenotypic drug discovery. So we can identify the target based and phenotypic drug discovery. AI, artificial intelligence, deals with biomedical, clinical, and patient data. For the repurposing programs, artificial intelligence can be used. Artificial intelligence are used for the biomarkers development, and it can use to analyze research literatures, publications, patents to get the information for further executing our research. And this I have given the top 10 hot artificial intelligence technologies, but many more technologies are there. These are all the most important technologies. That's why I have given you here. 1 minute. Okay, so these are the natural language generation through algorithm. So background of all the technologies is development of the algorithm. Natural language generation, natural language understanding, and speech recognition, machine learning, deep learning, virtual agents, expert systems, decision management, and robotics. So these are some of the text analytics, text mining, text analytics. These are some of the very important technologies. This is a combined one. Artificial intelligence is not a separate one. It's a various combined technologies. It's generally called as a artificial intelligence. So this is very important because researchers can say that is artificial intelligence should be used in the development of pharmaceuticals or drug molecules or identification of protein or validation of protein. But what about the practicals? In practical, what is the thing going on in the pharmaceutical companies? Yes, the pharmaceutical companies is utilizing the AI technologies. It has invested much amount for the development of the new molecules using the AI technologies or drug repurposing using the AI technologies. So these are the top 10 pharma companies. These are all using the technology for developing the new molecules or repurposing the molecules. Novartis, Pfizer, Roche, Sanofi, Merck & Co, Johnson & Johnson, GSK, AstraZeneca, Gilead Sciences, and Takeda. So these are the companies are utilizing or investing their fund for developing new molecules. So pharma industries in collaboration with the AI-driven companies or startups, new startups. So I would like, because in the previous slide I have shown you, the companies is investing much amount for development of their new molecule. So here in the last 2017, in June 2017, the Genentech Research is a AI-driven solution, in partnership with the GNS Healthcare. So they have made a collaboration to identify and validate the novel cancer drug targets using the company's proprietary causal machine learning and simulation AI platform. So they are making their collaboration to identify the new drug target, that is a novel drug, cancer drug target using the AI simulations. One more AI-driven company, Berg Major, Berg, not a major, it's a Berg is a AI-driven company. It collaborated with the major pharma. It has a major pharma collaboration with AstraZeneca, focusing on identification and evaluation of novel approaches to treat the Parkinsonism disease and other neurological disorders. AstraZeneca is an international company. Similarly, GNS is healthcare is a renowned international company. So they are investing their amount and making the collaboration for developing a new protein molecule and new drug molecule. Similarly, GSK has signed with the UK-based AI-driven company Exscientia with the amount of $43 million. It has made the collaboration to identify the small molecules for 10 selected targets across undisclosed therapeutic areas. The same GSK has made another one has made collaboration with another one company. This is a Baltimore-based AI-driven company that is called In Silico Medicine. So the purpose is to explore how In Silico's artificial intelligence capability can help in the identification of novel biological target and mechanism, that is the pathways of the interest to GS. Another one company, Santen Pharmaceuticals, is a Japanese leader, is a Japanese worldwide leader in the ophthalmic field. They entered in the strategic research collaboration where tools are to model the whole biological systems. It's not a particular protein, particular target. To model a whole biological systems to identify novel pathways, targets, and biomarkers. Takeda is another one worldwide company. It announced a multi-year research partnership with AI-driven drug design company called Numerate to develop new clinical candidates in oncology, gastroenterology, and central nervous system disorders. So this information gives you the it is insisted you the significance of the AI-driven in the pharmaceutical research. Biomarkers. Why I am showing the biomarker? So in the AI-driven companies, the main indicator for identifying the protein molecule is a biomarker. Biomarker is a substance or structure or a process. Whatever may is a substance or structure or process that can be measured in the body or its products and influence or predict the incidence of the outcome of the disease. So this is a clinical indicator. Biomarker is a clinical indicator. Simple example, creatinine level, if the creatinine level is increased in the diabetes, creatinine, it indicates the blood glucose level is increased. Here the creatinine is an indicator. So here in the first case, it is used as a prognosis agent. Before coming the diseases, it can forecast. So it can forecast the disease process. Understanding the relationship between the measurable biological processes and for deepening our understanding of normal healthy physiology. That is, that is a biomarker is used as an indicator in the pharmacological activity, normal physiological activity, and pathological condition, even in the pathological condition. Here, it gives some idea about the relationship between the measurable biological process for deepening our understanding of normal healthy physiology. And biomarker offer potential and promises for, so it has a tremendous potential that transform the pharmaceutical research and development in drug discovery area. Biomarkers are measurable, is a measurable one, We can measure it in biological media such as human tissues, cells, or fluids. So this is my, and then now we will see the research part. This is a flow diagram of analysis of the procedure, how I have carried out my research. The research title was Identification of Hub Genes and Key Pathways Associated with the Co-Infection of Urinary Tract Infection and Diabetes via Integrated Bioinformatic Approaches. The first part, so before entering into the research, the very important parts is collection of literatures. We should know the information about, so what is our exact focus of the pathogenic condition. In this research, the exact focus of the pathogenic condition is the co-infection, that is highly the diabetic patients as a probability, as a maximum probability for getting the UTI infections. They are easily susceptible to the UTI infections. So our purpose is to identify the common genes available in the both the cases. In both the cases, UTI as well as the diabetes. So from once the common genes are identified, we can develop the drug target to, then we can match the drug process. Because the patients are receiving the two modules of the drug materials. This is for the diabetes medicines is suggested. For the UTI medicines are suggested. So they are taking more level of the drug materials. It may produce a lot of side effects or drug-induced effects. That's why we wanted to identify the common modalities in the co-infection process. So for which we have collected the data. We have collected the data from various databases, that is OMIM, GWAS, UniProt, NCBI, and we have text mined using the R algorithm. So we have text mined. So we have given the code words and developed the algorithm. So we have collected a lot of genes from the various literatures. This is done separately. That is for diabetes, we have done separately. For UTI, we have done separately. And finally, we have collected the various n number of genes for diabetes, n number of genes for the UTI. And once it is collected, it has to be integrated. That is, the diabetes genes and UTI genes has to be integrated. The redundant genes has to be removed. The names of the redundant genes has to be removed. And finally, it has to be subjected for some overlapping analysis, identification of the overlapping gene. What are the common genes? So the separately collected genes are integrated and then it's subjected for the overlapping process to identify the common genes. Once the common genes are identified, so functional enrichment analysis has to be carried out. Functional enrichment analysis, that is, it means it is highly related to the gene ontology studies. It is highly related to gene ontology, that is cellular function, molecular function, and biological process. And finally, that is separate one, that is biological pathway. So functional enrichment study has to be carried out. And once the functional enrichment has completed, for all the common genes, so what are all the network among the common genes? The network among the common genes are proteins. It might be a gene or gene products. So what is the relationship among the gene products has to be identified. Once the protein-protein interaction is developed, and finally, hub genes. What are the most important genes? It might be a single gene may be connected with, it has a relationship with various genes. A single gene may be connected with various genes. That is called hub genes or biomarker. It has to be identified. This is the part one research. Once the biomarker is identified, the protein has to be developed. That is a target has to be developed. Target and substrate has to be identified. Then, as usual, screening of small molecules, molecular docking, molecular simulation, and other studies has to be carried out. This is to identify the identification of potential. Finally, we can identify the potential inhibitor for the co-infections. The next one, this is our study methodology, and we have collected the diabetes gene. Here we have collected the UTI genes. Both are integrated, and once it is integrated, it has to be subjected to the overlapping process. Once the overlapping process is completed, it has to be examined for its interaction network among all the overlapping genes. The protein-protein interaction was constructed. This is a separate software called string. And another one, finally, the evaluations. The protein-protein interaction has to be evaluated, and we can identify the hub genes. So here we have used the various databases and data sets. I mean, UniProt, various data sets we have used. And for overlapping, we have used the Venn diagram. And once it is the Venn diagram, once we identified the overlapping gene, it has to be subjected to the gene ontology study by using the Enrichment method. And finally, after the gene enrichment process, it has to be subjected to the protein-protein interaction. Finally, it has to be validated using the Cytoscape. The protein-protein interaction can be constructed using the string software. Everything is a online software, and there is no paid one. And finally, we used the Cytoscape for the evaluation and validation process. So this is some Geo, UniProt, GWAS, GenBank, PubMed. These are all the various databases, online databases, free databases available. Now we can utilize the gene. We can utilize to collect our data sets based on our pathogenic conditions, based on our research focused pathogenic conditions. In the Geo, we can get more high throughput gene expression and other function and microarray process and other functional genomic data sets. Various types of data sets we can get from every, in the types of data sets will differ from the database to database. This is a UniProt. UniProt are high quality and freely accessible resources of protein sequence and functional information. We can get with the protein sequence. It's not only as a gene. So what are the protein sequence of the particular gene? And GWAS, it gives the findings from genetics association studies. It's highly related with the genetic association studies, both large and small level. And GenBank is a open access, annotated collection of all publicly available nucleotide sequence and their protein translations. And PubMed is all the publications. It is stored in the PubMed. We can get the information from the PubMed. So information that we can collect the data set even from the PubMed literature. And how this is our chosen approach. Already I explained to you. So this is Geo, and within the Geo there are the Geo2R. This is one tool in the Geo2R. This Geo2R is used to compare that is 2 or more groups of the samples in order to identify the differentially expressed genes. What I have collected for my research is completely all the genes are not differentially expressed. Only I have collected the reported genes, but what are the data sets I collected using the Geo2R is the Geo2R is a differentially expressed genes. And the functional enrichment analysis and the interaction network and analysis of genes and proteins. What are the genes and the proteins are interconnected genes and proteins are identified using the FunRich software. And this is a string aims to collect and integrate this information by consolidating known and predicted protein-protein association data for a large number of organisms and visualizing the molecular interaction networks and biological pathways and integrating these networks with annotations, gene expression profiles, and other data. So here I am showing you some method of search in the gene expression omnibus that is called Geo. So here there is some, so in this place, it is hidden here, there is query words, it will ask some query words. So I enter the diabetes. The diabetes, it shows there is 27,500 data sets and 37,600 databases. I require only the data sets. That's why I click that. Then So the second page will appear like that. The important thing here is it has various filters. This filter is hidden here. So this part is not visible here. These are all called as filters. Here in the right side, there is a filter whether what type of organisms, whether we are searching for animal origin or Homo sapiens or for the human origin. If we want only the human origin, we have to enter the Homo sapiens. Then we can get so here this is study type expression of profiling by array, methylation profiling by which one we require. So everything, or if we want to enter something in the customize, we can enter our details. So by this way we can collect the data. This is another one software, the NCBI available, OMIM software. OMIM is a database, not a software. It's a database. We can collect the detail here by searching here, by entering our query word. We can collect the data sets here. So once by this study, by this using the database, we and my team has collected the around 1,600 gene. That is not 1,594 gene for diabetes and 294 genes for the UTI. So we have collected separately, and for the next study we have integrated. So we have integrated this one. So here 1,594 and 294. Okay. So here, so this is a very common genes in both the cases. In both the cases, this is a common genes. What is the purpose of the Venn diagram? It uses a circle to show the relationship among the things. So what is the relationship between the diabetes and UTI? The relationship is based on the sharing of the traits by the biomarkers. These 64 biomarkers are sharing their traits with the diabetes as well as UTI. So this is these commonalities has to be identified. Circles that overlap have a commonality, while circles that do not overlap do not share those traits. Venn diagrams help to visualize and represent the similarities and differences between the two concepts. So here I will show you. This is a similarity. The 64 is similarity for the diabetes and UTI. This is a dissimilar biomarkers 1594 and 294. This is a dissimilar biomarkers. The number of overlapping genes among the diabetes and UTI genes. This is the 64 numbers are common genes for the UTI and the diabetes. This orange color is a common modalities. And once we identify the common genes, the gene has to be clustered. That is, the similar genes has to be clustered. That is called functional enrichment analysis. For clustering, there are so many softwares available. The best software, David, is one of the software. Here I utilize the FunBridge. FunBridge is a standalone functional enrichment and network analysis tool. Functional enrichment analysis is a method to identify the classes of genes or proteins. That is, a similar genes or similar classes of proteins are separated. It might be based on the disease or based on the mechanism that are overrepresented in a large set of genes or proteins and may have an association with the disease phenotypes. So the overrepresented genes are having the association with the disease phenotype, development of the disease. The molecular functions, cellular locations, and processes of gene products may carry up. So what is the cellular location, where the exact cell is located, and what is the activity of the cell in the molecules, and how the process is developed. So this can be analyzed. This can be analyzed in the functional enrichment analysis. So another one term for the functional enrichment is the gene ontology. It means that is a similar thing, representation of the, it entirely represents the gene and gene products. So gene ontology means representation of gene and gene products. And this is a very important thing. I would like to show you with some examples. Gene ontology study. This is a gene ontology study. The gene ontology is generally related with the cellular function and molecular function and biological process. In the cellular compound, the plasma membrane, this is the plasma membrane, it has here around 40.3% biomarkers are available in the proteins are available in the plasma membrane. 40.3% of the genes or biomarkers are located in the plasma membrane, and 47% were identified to participate in the nucleus, and that is here in the nucleus. Here there is a 47%. Here there is almost 46.8. So 47% are participate in the nucleus. Almost 29 genes are present, and 41% of these genes are presented. Our gene products are located in the cytoplasm. The major that is where is the origin of the disease? We can identify that by knowing the information of the cellular mechanisms. We can identify the origin of the diseases. And similarly, 32% of the genes are located in the exosomes. Here, 32%, 0.3% are located in the exosomes, and 29.5% of genes are located in the extracellular. Here, 29% almost are located in the extracellular region. So now we identified that where exactly the genes are located. That is, these genes are common for UTA and diabetes. All the 64 genes locations we have identified. And what is its active? How it execute? So what type of activity? So now here there is a 1.9% and 46% genes are available in the nucleus. Similarly, in the exosomes, so many number of nucleus are available. And what is its molecular function? What type of activity it possesses? So here in the molecular function, here the RNA binding activity, DNA repair protein. These are DNA repair protein and metallopeptidase, growth factor. These are all the activity is common for the UTI as well as the diabetes. So what we are writing, this is not the transcription factor activity is common for both the things. And the voltage gate ion channel activity, growth factor activity, these are all a very common thing for the diabetes as well as the UTI. And biological process for the common genes. So what are the biological process is a very important term. Here it has a definite time. The biological process has a definite time for the beginning and ending of its process. There is a definite for transport of some molecule means it has a definite time. And energy pathways, protein metabolism, and immune response. When the immune will respond, give the response. If there is any antigen or foreign body enters, it will respond it. But within the particular time, it will be over. And cell communication, DNA repairing, and signal transduction. So biological process is, say for example, is genesis of any organ. Is the genesis of any. It might be a tumor, or it might be in some indicator, that is a biomarker, a pathogenic indicator. So this process is called biological process. What type of changes it gives? So we can identify the pathogenic condition based on these three studies, that is a cellular mechanism, molecular function, and biological process for the common genes. And next one is the biological pathway. So what is biological pathway? Now we identified where is the gene located and what type of activities it is going on and what type of changes is produced in the body. That is a biological process. Is there any changes? Is there deviation from the normal physiology to abnormal conditions? So this is a biological process. Now our question is, this is all the mechanism is not known to us. So in this condition, AI will be useful to identify the exact biological pathway. So it will give us the answer what type of mechanisms involved. So biological pathway analysis here, this is some indicator for the biological pathway. It's a complicated one. There is biological pathway, it's not a single gene, it's not created by a single gene. It's a very complicated, multiple genes are involved, multiple gene products are involved. So here biological pathway, in-depth and contextualized findings to help to understand the mechanism of disease in question. So this is it gives us that is a in-depth analysis. It gives a new insight to our questions and identification of what are the genes involved and proteins associated and etiology of the specific disease. So we can exactly filter the unwanted genes in the cases, and what are the genes actively involved in the pathogenic conditions can be identified by knowing the biological pathway. Then prediction of drug targets, what is the, that is if we know the cellular locations and what type of activity and where is the process is, what type of changes is located, so we can identify the biomarker. Based on the biomarker, we can identify the protein target, understand how to intervene therapeutically in disease process, and finally conduct the target literature searches. So this is based on the information obtained from the biological pathway, we can know all these 5 things. And biological pathway, so this is a integration of the previous 3. This is a integration. Biological pathway analysis is that is after uploading the 64 genes, that is a common genes in the fund. This is also carried out. 1 minute. Sorry for the interruption. Okay. Biological pathway is after identification of the disease. What are the mechanisms involved can be carried out using this biological pathway. This pathway is common for both the disease. Here the insulin pathway and VGEF, vascular endothelial growth factor. So can be, this is common in both the cases, that is in diabetes as well as the UTI. And another important thing is urokinase type plasminogen activator. This is involved in both the cases. And proteoglycan syndicon mediator signaling events. So these are all the events, or these are all the pathways which can be identified using the that is AA technologies. But these all the pathways are involved in both the diseases. So this is how the co-infection is developed. Can be this is the answer for the our question of how the co-infection is developed. What are the pathways involved? Now we identify the pathway. So based on the information, we can move on to the protein-protein interaction studies. Protein-protein interaction studies. So here, this protein-protein interaction, there is here, this is an online tool. It is called, generally it is called as here string search tool for the retrieval of interacting genes. Is here retrieval of interacting genes. We have identified 64 common genes, and gene enrichment analysis can also be carried out. And we have identified the what are the genes, similar type of genes are involved, and what are the genes are sharing their activity in the process of development of the pathogenic conditions or physiological conditions. So everything we can identify now. So it is a time to identify what kind of relationship all the 64 gene possess. What kind of relationship all the 64 gene possess? This is a very important thing. So this tool is an online software, string protein-protein interaction network, functional enrichment analysis, and it is a biological database and web resource for known and predicted PPAs, protein-protein interactions. We construct a network for PPAs for 64 overlapping genes. All the 64 overlapping genes are identified. The string database contains information from numerous sources. It's not in the, that is in the background, it collects the information from the numerous sources. It's not from the a single source. It collects the information from the Geo, it collects the information from the Unipro, it collects the information from the cake pathways and PubMed, and Gen Bank and GWA. So it collects information from all those things, including all the experimental data and computational prediction methods and public text collections. And so all the information it retrieved from all the sources. It is freely accessible and it is regularly updated. The resource also serves to highlight the functional enrichment in user-provided list of the protein using a number of functional classification systems, what I told you. So this is a regularly updated one. So it's not a single, it's a fixed one. So after getting the there is a newer information, the data will be updated in the string database. So many people are raising your hands. I will answer you once the talk is over. Okay. The PPI network. This is a construction of PPI network for all the 64 genes. So how all the 64 genes are, whether it is in the relation or not. So almost all the genes are having the relation with each other. These two are somehow separated, and this gene and this gene are having the a single gene. This is a single gene is connected with many gene products or genes. So this is a candidate molecule. These candidate molecules are connected with many of the nearby or neighborhood genes. Okay, so this PPA network constructed by this is to identify whether all the 64 genes are having the are sharing the common traits or not. So it almost proves that all the genes are sharing the common traits, but very few genes are sharing that is very closely than others. Then the rest of the genes, very few genes are having a high correlation with the neighboring genes. So this string analysis is having the it is used to create the protein-protein interaction map. It is used to create the protein-protein interaction map. Here we can identify whether all the proteins are having the interactions or not. So this is another one representation pattern. So this is the same string. This is another one representation here. A single gene. This is a candidate gene. It has interaction with many more molecules. Here a single cluster. It is called as a cluster. It shares with many more interaction. But if you look at why I show you that, I told you in the string it collects the information from the various data. So various databases. So this is these are all the interactions between the one molecule to other molecule. This is the background of the genes. This is the background of the string protein-protein interaction map. So it has information from the various databases. It retrieves the information from the various databases. And finally, this this is another one online tool. This online tool is used to validate and evaluate the protein-protein interactions. This online tool is used to evaluate or validate the protein-protein interaction studies of string. What we have obtained the results from the string, the detail has to be fed into the Cytoscape. So based on the results of the string, we can get the hub genes. Hub genes are like a biomarker. A single candidate molecule is having the high correlation with the neighborhood molecule or nearby molecule. This can be hub genes are identified using the Cytoscape. In the Cytoscape is a common name. In the Cytoscape, it has it's a plugin software. It has many software inside the Cytoscape. So here in this study, we have utilized the there is a tools. There is a two types of tools I have utilized. One is CytoHubba and another one is a CytoGlue. The Cytoscape software was applied to visualize a protein-protein interaction relationship network and analyze the hub proteins and which are important nodes with many interaction. These are all the very important nodes with the many interaction patterns. Okay. So in the CytoHubba application, there are the various factors to be parameters to be employed. We can employ various parameters, but I have chosen in my study only the 5 parameters, that is degree and EPC and MCC, MNC and bottleneck is one of the another one parameter. So the interesting genes derived from these 5 algorithms encode the core proteins and may represent the key candidate genes with the important biological regulatory functions. We will see you one by one. So this is this IL-10 and VEGFA. That is a vascular endothelial growth factor A and interleukin 10. So this is the most significant gene or gene products in the 64 common genes. Among the 64 common genes, these 2 are most significant. These 2 are generally called as it poses a high correlation with all the neighborhood genes, And rest of then next to the IL-10 and VEGFA, TLR2 and CRP, and C-reactive protein and toll-like receptors. Okay, TLR2, TLR4, these are all having the good relationship. And next to the IL-10 and VEGFA, and this endothelial factor, angiotensin converting enzyme, S100A9 and CD9 LCK. So these are all also having the good interaction with the other molecule out of 64 common genes. So there are 64 common genes. In the 64 common genes, these 2 are highly is ranked as a topper row 2, and other 3 are TLR4, TLR2, and CRP. So 5 genes, and these are also the overall. This is top 10 genes I have selected. These are first 2 and 3, 4, 5, and rest of the genes are in the next rank. Okay, this is used to identify them most closeness of the that is a candidate genes. This is used to identify the candidate genes. Candidate genes means a single gene will have the multiple interactions and multiple connections between the one molecule, that is what it is called as a gene to gene product, one gene to another gene, or one gene product to another gene product. So it shows a very closeness among the interactions. Rather than the other gene products or other genes, these 10 gene products or these 10 genes shows a very close relationship. So these 10 genes have been chosen as first of all, these 10 genes we have chosen as hub genes for our next study. This is a cytoglue. Cytoglue is so far we have conducted the overlapping study and gene enrichment study and protein-protein interaction and validation of the protein-protein interaction using the cytohubba. The cytohubba, we have identified the 10 common genes, and these 10 out of 64 genes, these 10 common genes are generally called as hub genes. And among the 10 common genes, I have chosen only the 5 genes. It's a top 5 genes for our next study, for the next process of the study, for developing the protein molecule, for developing the protein, and to identify the exact lead molecule. And using this cytoglue GO, and application in Cytoscape was employed to identify the significant pathways and the similarities in terms of associated genes in complex flow works. So what are all the genes is like a clustering process. By integrating the cellular function, molecular function, biological process, and biological pathway, and finally finding the similarity of the genes in both the cases, in both the diseases. That is, we can identify using the cytoglue GO. This glue GO was performed to integrate the gene ontology terms as well as KEK pathways and creates a functionally organized gene ontology and pathway term network for the identified hub genes. So this is the gene process. This what are the genes we have chosen here? It indicates this is 100 and TGF involved, and these are the IL-10 here and CD10 here, and so many gene processes are involved. Here it indicates that these genes are responsible for various diseases. What are the other diseases are correlated with the particular genes? It gives information not only the diabetes and UTI, it also gives the other information that is other than UTI and diabetes. These genes will responsible for various diseases. So this information can also be received by the method. So here I wanted to give you one more example that in the previous case, that is bottleneck in the cytoscape, here I have chosen the 5 parameters, that is degree, MNC, MCC, and bottleneck, and betweenness. Betweenness is here one of the parameters. So what is the why I have chosen the particular parameters? Just I wanted to give you 2 examples for the betweenness and bottleneck. Betweenness, it means is a key connector. Betweenness means a key connector. So this is a LCK. It is a key connector among all the molecules. The shortest path is very important in identifying the betweenness. The case of IL-10 and VECFA, the IL-10 has a very shortest path between the other genes. Similarly, VECFA is having the shortest and longest path, but it has a more connection between the neighborhood gene molecule, between the neighborhood gene molecules. So this betweenness, it gives some idea. It means 2 key connectors are exhibiting the high betweenness, the interacting partners, and share similar functions. If the betweenness, the distance is very short, it means it has a similar function. It has a similar functions. And this betweenness will also be useful in controlling the communication among the other node in the network. So it is very useful. If the path is very less, it will be very useful to control the communication with the other networks. So within the shortest path, it will communicate all the informations rather than the longest path. And another one is bottleneck interaction networks. So the bottleneck interaction networks, it is I have find a surprise finding. There is a distinction between the interaction of the proteins, and other thing is regulatory interactions. That is this is the protein-protein interaction. And another one is a regulatory interactions. So both are having some differences. It doesn't possess a similarity, it possesses some differences. This is our different finding in the research. So among the 64 molecule, using the 5 parameters, I have chosen only the first of all 10 molecules I have chosen, 10 gene products I have chosen. And using the gene products, I have identified clustered. That is, I have identified or clustered the similar pattern gene and disease process, and similar mechanism of action of the gene. Similarity of the genes have been identified. This is a similarity. I told you. So in the previous case, it's not very clear. So this is different color. This is different color. These are all different color. And here I have clustered. So this is a similar activity. The 3 clusters, we have obtained the 3 clusters. All the genes are comes under the 3 clusters. The majority of the genes are possess this type of diseases. This is common for both the diseases. This is various signaling pathway. This is a pathways. What are the various pathways involved in the disease process? So macrophage differentiation, lipopolysaccharide mediated signaling pathway. These are all very important for diabetes as well as UTA and for other diseases. Not only these diseases, this is common for rheumatoid arthritis and inflammatory bowel diseases. So it gives 360-degree information about the genes and gene products. So that's all about the part 1 of the identification of hub genes using the bioinformatic approaches. And part 2 is totally different. So once the biomarker and the process is identified, once the biomarker and the process are identified, we will go for the protein, that is a target identification, target validation, and drug discovery process, and similarly lead identification and the drug interaction process, drug and protein interaction process. So and then finally it will go to the clinical trials and phase 1, phase 2, phase 3, and phase 4. And phase 1, phase 2, phase 3 is before releasing the drugs, it has to be carried out. And phase 4 is the post-marketing surveillance after the release of the drug. This trial has to be carried out. Okay. And next one is I conclude the part 1. This is a part 1 conclusion. So what is the benefit of identifying the biomarkers or using the bioinformatic, integrated bioinformatic approaches with the background of artificial intelligence? The artificial intelligence gives a complete details about the origin, function, and development of the process, and what are the mechanisms. So everything we can get it clearly using the artificial intelligence. So it increases the efficiency and reducing the cost of the drug development process. And it developed the predictive biomarkers. So not only for pathogenic condition, for the prognosis conditions, that is before coming the diseases, we can able to identify. It developed the predictive biomarkers to a molecular targeted therapy before the drug is tested in humans. Radiology, disease diagnosis, gene therapy, finding drug interactions, and biopharmaceuticals. Not only even the biomedical field, in all the medical field, this artificial intelligence is mainly used. Developing the electronic equipments, even in the COVID conditions, so many electronic equipments are developed. The background is artificial intelligence and IoT, Internet of Things, also used. And this process improve the understanding of process of pathogenesis and underlying molecular events for the precise treatment. And we can understand the gene ontology, protein network, and protein-protein interaction and validation of protein can be carried out without collecting the literature from the hard copies. Everything can be carried out in the databases. By uploading our details, it could be accomplished in a shorter duration. That is very important. Within a shorter duration, we can accomplish this study. Finding therapeutic effect of toxicity, profile of drugs, prediction of structure, bioactivity, and mode of action of drug, selection of population for clinical trials. So these are all the advantages of the artificial intelligence in the field of in the field of drug development process. So the integrated bioinformatic drug discovery process was very useful and reduce the time and give the 360-degree information about the drug target and pathogenic condition and physiological condition and pharmacological process. And one more important thing, this is a teamwork. A single person cannot carry it out because I have given you the lot of databases. All the databases are created by some people. They are not related to our field. There may be some other database specialist or data analysis automation and molecular biologist, mathematician, data processing specialist, statistician, and doctors, pathologists, oncologists, IT infrastructure, and civil. So every people are involved in the teamwork of bioinformatics setup. So finally, I would like to thank Professor Dr. G. Narahari Shastri. He is DST JC Bose National Fellow. Currently, he is Director of CSIR Northeast Institute of Science and Technology. He has given me a wonderful opportunity to learn the technique. And I thank my D3 research group, Dr. Uma Maheshwari, Dr. Yesh Lakshmana Prabhu, Dr. N. Irfan, and Mr. Balasubramanian, Dr. Balasubramanian. My collaborators, Gesh Duble, IIT Chennai, and Dr. Sriram Bits Pilani, Dr. T. Velmurugan, University of Madras, Chennai, Dr. Nandakumar, Manipal University, Manipal, Chennai. So we have collaborated for various publications and research activities and research projects. These are all my protein interaction. This is a drug-protein interaction. This is not at all a biomarker study. We have identified a new drug target. It's a not a new, it's a novel drug target for the mutated UTA infections. That is, we have identified the new drug target for the fluoroquinolone resistant E. coli. So this is research, and this is 3D structure interactions and qualitative structure toxicity studies of tyrosine derivatives for the inflammation treatment. And this is my collaboration with IIT Chennai. This is SAHA for selective histone 8 inhibitors. This is my patent. So in my introduction, they have given some information about the patent. This is one of the research, and this is my patent. And these are the publications published by the Natural Products Drug Development, Computer Applications Drug Discovery and Development. Thank you. Thank you, and thank you for giving me the wonderful opportunity.
Thank you, madam.
Thank you, ma'am.
Immense sharing for your immense knowledge in the area of proteomics, genomics, and prognosis of disease, how bioinformatic tools helpful. Now I request participants to type your questions related to the presentation in the chat box so that madam will answer them.
Shailaja, already some questions are there.
Yeah, please, you read it, ma'am.
Pooja, open the chat box. I'm not able to see here.
Ma'am, yes, ma'am.
Just post the question.
The question is, could you please give any free databases available online for retrieval of ligands in bulk to check on a target?
Yeah, many databases. Zinc database is for mainly for the retrieval of ligands. Many more databases are available. Here, what I have given you is mainly for the not for ligands. That is for biomarker identification. For ligand identification is a separate one. In the best one is Zinc database. Many databases are available. You can type in the Google, you can get it.
Yes. Thank you, ma'am. Next question. How to develop training sets for artificial intelligence?
Training set for artificial intelligence.
Yes.
What he exactly want, I don't know exactly. Training set means he is asking the data sets. We need not train anything. Everything is already trained. It is in the, what are the details available in the databases is the background of the database is the artificial intelligence, machine learning, deep learning, everything is involved. We can upload our details and methodology and many filters are there. We have to select the filters and the method procedure has to be adapted so we can get the results.
Are PPI modeling and artificial neural networks same things? Or
yeah, almost same. Yeah, in the AA is an artificial ANN is a very old name. Now it is comes under the artificial intelligence. ANN is a predecessor for the AA. Deep learning.
Josh, why don't you share the chat box so that madam can also see the questions? No?
okay.
Madam, another question is, how does artificial intelligence generates the database? How reliable and accurate is artificial database?
Yeah, it's a right question. And the database is developed by some, already I told you, database developers are developing the databases. Even if you are much interested in this area, we can develop. If we know the technology of artificial intelligence, we can develop it. And regarding the confidence level, 100% we cannot sure, but there is this is the only technology. We cannot skip the artificial intelligence because we need to waste the time, investment of amount, everything, a manpower involved. That's why we are relying on the artificial intelligence. but almost more than 90% is believable based on the data sets available in the databases. The base material has to be curated one. So already everything has to be scientifically curated. If the information is curated, so everything will become true. Otherwise, it is very difficult.
Next question, ma'am. Can CAD and AI be collaborated? If so, how beneficial is it for CAD?
CAD and AI,
AI, ma'am. Artificial intelligence.
yeah, collaborated.
If so, how beneficial is it?
Uh, uh, because the technology process in the artificial intelligence, I told you robotics is a part of the artificial intelligence. CAD also comes under a part of the machine learning. Probably, I don't know exactly. It's a part of the machine learning. Already, this is in the background of the artificial intelligence. Technology is in the background of the artificial intelligence.
Last
Somebody
question.
asked, ma'am, somebody asked that if you are using any software such as Design Expert or JMP for my research work, is it also called the use of artificial intelligence?
Yeah, because yes, madam, all the softwares now because it has a library of molecules. This library of molecules is already stored. If it is in the online process, this is very important. Software means there might be a limited library molecules. It is already a fixed one. It cannot be updated time to time. But if it is in online always, it's a time to time updated one is comes under the artificial intelligence. I told you string is updated time to time, and all the databases are will be updated. Freely available databases are updated time to time. Means is based on artificial intelligence. Even the paid software, if it has the ability, if the company people is updating time to time, it means it's artificial intelligence. Hello, ma'am.
Yes, ma'am. Yes. Okay. Ma, next question.
Are these softwares licensed, ma'am? Is the license free?
Everything is free, madam. All are available in the net. That's why I have shown you this online software, freely available software. Paid also available, but for initial works, we can use the freely available software. So, what are the work I have presented here? All are from the free softwares. No one is paid.
Are these databases available, genuine and reliable?
The question already asked, ma'am. I told you. The question already asked.
Yes.
Similar kind of question already asked. It's a reliable one. It's a curator. It's a reliable one.
That's all. Thank you, ma'am.
Thank you, ma'am. Thank you so much.
Any questions from participants? Okay. Shailya. Okay, if no questions, we can conclude. Is Shailya is there?
How AI is different than machine learning? Somebody is asking.
Yes,
Machine
ma'am.
learning is part of the AI. Machine learning is part of the AI. In the slide, I
Yeah.
told you. So, what are the machine learning is part of the AI.
ఏంటే
Hello.
Hello, Pooja.
Ma'am, yes, ma'am.
మేడము అన్మ్యూట్ చేయాలంటమ్మా. మేడం నువ్వు శైలజ మేడం అన్మ్యూట్ చేస్తే వస్తారంట. ఎక్కడున్నారు మేడము? కనిపించట్లేదు నాకు. Because of that reason, she is facing some problem. She connected to some other network. She told like that.
Now I am able to know. Thank you.
You unmuted the madam?
Yes, Sridevi, I am here.
Okay. You can conclude, Shailya.
Okay. I would like to thank Professor Purat Chikodi madam for sharing her immense knowledge on artificial intelligence, role of artificial intelligence in genomics, proteomics, and prognosis of a disease. How it plays a role in detection of disease, the proteins related to detection of disease, and madam also explained various artificial intelligence related tools available in exploring data related to drug discovery and validation, that is disease madam also highlighted on different tools available explore artificial intelligence related to drug target identification and validation. I also thank the participants for their active participation in the faculty development program. Madam, I would like to thank you for sparing your time towards this FDP program. The session, I think Sridevi is the session.
Madam, my heartfelt thanks to you. So as soon as when I approached, although I don't know you, so as an unknown person also, you are readily accepted. Whenever I call you, you are very patiently answered, and you are given the suggestions how to do, and you are also given 1 or 2 resource persons name which are very helpful for us to conduct this FDP. So thank you, madam. Thank you very much for your session, and you are elaborated and elaborately explained everything on the artificial intelligence. So for that, we are very, very thankful, and many of the participants are also satisfied. They are posted in the chat box as very informative session this is. For that, we are very thankful, ma'am. So in future also, we are also expecting same thing. For any type of help we need, we will approach you, madam. Thank you. Thank you very much, ma'am.
Thank you, Dr. Sridevi. It's time to say, because whenever you approach me, you are politely and you asked me to participate in the event. It's my commitment as a researcher. It's my commitment to share the information. What are the things I have known has to be shared with others. That's why I participated. And thank you for giving me the wonderful opportunity, because persons from I saw the participants list all over India. Participants from all over India joined together. Yeah, it's I'm so glad to deliver my lecture within the people, with the people. Thank you so much. Thank you for giving me the wonderful opportunity.
Thank you, ma'am.
Whenever you call me, definitely I will support.
Thank you, madam. Thank you for your
concern.
concern.
Thank you, Dr. Shailaja. Thank you. Thank you.
Thank you, madam.
Thank you.
Really, the lecture is interesting. The genes related to, as you explained, overlapping genes between diabetes and UTI infections. I have never heard about this thing.
It's a new research. It's not known.
C-reactive protein and interleukin 10 role you explained very nicely, madam.
Yeah.
Interesting.
Thank you. Thank you.
Thank you, ma'am. Dear participants, our afternoon session will start at 3 o'clock, but please join at least 15 minutes before. Okay. So with this, our first session is over, and sharp 2:45, kindly join, and by that way, sharply 3 o'clock, we will start the session. Thank you.
Thank you. Thank you.
Thank you, ma'am.
I will take leave. Thank you.
Thank you, ma'am.
Thank you.
Once again.
Thank you.